Hello guys, good to see you again. Welcome to today's lesson. We are discussing anatomy and physiology. Our topic today is embryology. As we discuss about embryology, there are some basic concepts that we need to understand. One of them is the process by which gametes form. This is a process called gametogenesis. There are two types of gametes, the female gamete and the male gamete. We call the male gametes spermatozoa and we call the female gametes ova. Now, formation of sperms, that's spermatogenesis, usually occurs in the testes, whereas formation of ova, process called oogenesis, occur in the ovaries. Now let's look at the process of spermatogenesis at length, and then later we'll look at oogenesis. Now this process occurs in the seminiferous tubules, which actually form the bulk of each testis. Now the process begins usually at puberty. After one reaches puberty, sperms are produced constantly throughout a man's life. Men do not undergo menopause as it is with females. Now, the amount of sperms that a man is able to produce, or the number of spermatozoa that one produces in a single ejaculation, is normally called a sperm count. It is important to note that sperm count basically reduces with time. Once you hit the age of 35, the amount of sperms tend to reproduce, and that accounts for cases of impotency as we tend to age. Now, spermatogenesis begins with mitosis. That's division of the original mother cell, what we call a spermatogonia, into two daughter cells. Therefore, the initial mother cell, what we call the spermatogonia, is normally a diploid cell, and it undergoes mitosis to produce two daughter cells, which are also diploid. I'm sure you understand the process of mitosis. We discussed it much earlier. Now, I would need to note that mature gametes are, however, haploid and not diploid. That means that the daughter cells of spermatogonia, or the cells that result from the initial mitotic division of a spermatogonium, must undergo a second cycle of division, which then will meiosis. So that at the end of the day, we have got sperm cells which are haploid rather than diploid. So basically, this is the stages through which spermatogenesis take place. So this is the original mother cell, what we're calling a spermatogonium, which is diploid. This cell undergoes one mitotic division to give you two daughter cells, both of which are diploid, as I've said earlier. Now, one of the diploid daughter cells develops into another spermatogonium, whereas the second daughter cell, what we now call a primary spermatocyte, will undergo the first meiotic division to give us two daughter cells which will be haploid. We call these haploid daughter cells secondary spermatocytes. Each of the secondary spermatocytes will then undergo meiosis II to form what we call spermatids. Spermatids are actually primitive forms of sperm cells, which will undergo more development and the growth through process we call spermiogenesis to form mature sperm cells. Generally, one spermatogonium undergoes first mitosis and then meiosis 1 and meiosis 2 to give us a total of 4 sperm cells. So basically, that's how spermatogenesis takes place. It's important that you also understand the structures of a sperm cell, because this will be important in understanding how the sperm cell functions. The sperm cell is actually a modified cell with a prominent head, and that has got a mid piece and a long tail. We also got an end piece at the end of the tail. Within the head region, we have got a prominent nucleus, which is enclosed by a plasma membrane, and also on the one side I've got an acrosome, which is basically a structure that contains enzymes whose role we'll be discussing much later. Within the mid piece, we have got a lot of mitochondria, which is an energy store to be able to facilitate the movement of the sperm cell through the uterine tubes in order to fertilize an ovum. Okay, that's the basic structure of a sperm cell. Now, spermatogenesis or sperm formation, as it were, is controlled by a number of hormones. The process at puberty is actually initiated by a release of a hormone called gonadotropin releasing hormone, generally GnRH. This hormone is secreted by the hypothalamus. Now, gonadotropin releasing hormone in turn stimulates the release of another hormone called luteinizing hormone and also follicle stimulating hormone. These two hormones are released or secreted by the anterior lobe of the pituitary gland. Now, the role of luteinizing hormone during spermatogenesis is to stimulate the interstitial cells of Leydig in the testes to secrete testosterone. Testosterone is a very, very important hormone in spermatogenesis and also plays a very, very key role in the onset of a male secondary sexual characteristics. Testosterone in conjunction with follicle stimulating hormone will initiate spermatogenesis or division of the spermatogonia by mitosis, which will eventually lead to formation of sperm cells. Once the process of spermatogenesis is initiated, testosterone also plays an important role in maintaining that process up to the end or up to complete formation of sperm cells in the testes. Now, an increase in the level of testosterone somehow inhibits the release of gonadotropin releasing hormone by the hypothalamus. That means that testosterone is able to control the rate of sperm formation. So that if the rate is very, very high due to a release of a lot of testosterone, the same testosterone at high levels will inhibit gonadotropin releasing hormone and thereby reducing the rate of sperm formation. Testosterone, we need to note that it also acts directly on the anterior pituitary gland to reduce secretion of luteinizing hormone. Basically, that is how hormones play a role in regulating the level, regulating the process of spermatogenesis. Thank you for attending today's lesson. We shall continue and discuss more about gametogenesis in our next session. Please look out for the next video on the process of ova formation, or rather oogenesis. Have a great day.